Hi, Imad. Thanks so much for joining me on this episode of Concentrating on Chromatography. I really appreciate it.
Thank you very much for the invitation. It's a pleasure to talk to you.
Just to start us off, would you be able to introduce yourself and tell us a little bit about your background?
Sure. My name is Imad Haidar Ahmad. My background during my PhD is in polymer analysis. I did postdoc with Peter Carr on chromatographic separations and fundamentals of chromatography. Then I worked at Novartis for a few years, focusing on the drug product side, method development, validation, close to late stage. Then I moved to Merck, where I worked on drug substance for some time. Then I moved to the specialty groups, the chromatography separation, as well as purification. Spent around 5 years in that area. And a few years ago, I moved to Amgen. I'm currently leading the separations team in discovery phase, which is very different from what I've done before. And currently, the group is mainly focusing in supporting discovery phase, a lot of purification, chiral, achiral, method development, analysis, physical chemical characterization. I would say, in general, the experience is in the separation side as well as the characterization from my PhD.
How did you first get interested in chromatography and what drew you towards analytical chemistry as a career path?
Thank you for the question. So my interest in analytical chemistry started when I was an undergrad. I did a mini project in my last year about analysis of additives in bread and seeing all the techniques you could use to analyze the content of bread was really amazing and got me interested in general in analytical chemistry. So when I pursued my PhD, I worked with Dr. Andres Striegel focused on polymer analysis, structural property relationships. And as I was finishing my PhD, I got really interested in some of the 2D-LC work that Peter Schoenmakers and Harbash was doing, where you could look at all the types of heterogeneities you have in these polymers. And the only way you could do it is when you couple size exclusion chromatography, which I was already doing in my PhD, with another chromatographic technique, say LC. So that's where the interest in chromatography was triggered. And that's when I did my postdoc with Peter Carr, where I learned about the fundamentals of chromatography. And from there, it was more and more chromatography and also style. From time to time, we still do some large molecules polymer analysis.
My understanding is that your work sits at a very interesting intersection between chemistry, software, and pharmaceutical development. Did you anticipate that this is what your career would look like when you were doing your undergrad, or did this kind of evolve unexpectedly?
It definitely evolved. It was, I would say, a gradual change with time, and I was not expecting I'll be in this field. When I was doing my undergrad, I was really impressed by the inorganic chemistry lab, also the course where you make all of these complexes, like you make crystals of all different types and colors. That lab was really fascinating. Compared to the organic chemistry, still, I like that course and lab, but it's always a white powder. Sometimes if you mess the reaction, it's a little bit yellowish. But I had really tendency to go pursue a degree in organic chemistry until I did in my final year a mini project where we looked at analysis of bread. That's when I was introduced to the analytical chemistry in the lab. It was really fascinating. And also taking the advanced analytical chemistry, seeing all the nice tools, powerful techniques you could use in that regard, triggered my curiosity in analytical chemistry. And as I mentioned, I learned later on the chromatography part. So that was a smooth transition into analytical chemistry, chromatography. And when I started in industry, you are challenged with problems. You have to find solutions. You have to think out of the box. And one of the largest problems you encounter in pharmaceutical industry is method development. And you have to be creative to meet the goal of a robust method, especially in my first job at Novartis, where we're close to late stage. You really need a robust method that you could transfer, you could validate with no problems, put it in the hands of any analyst would not break. And that's how they transition into implementing more of these tools and combining the knowledge from my PhD, postdoc, and pharma industry.
And most of the listeners of the Concentrating on Chromatography podcast are academics. They could be undergraduates or further along in their journey. I think they would be very interested to hear from you what a typical day looks like for someone leading a synthetic separations team.
First of all, I have to clarify that it varies from one position to another on which stage of drug development you are. Currently, we're in the discovery, so speed is the key. We really need to act fast because we're working with thousands of molecules and we need to push forward what's the best candidate to process development. Now, on daily basis, we receive requests from our partners in medicinal chemistry, also other departments. The goal is to be able to prioritize these requests within the same projects, among different projects, and push forward what's urgent. So we need to get that done as fast as possible. Within the group, there's for every workflow, there's someone who's in charge of making sure we deliver what we need to deliver. So besides prioritization, tracking requests on daily basis, communicating with partners, making sure that we are on track, we deliver what they need us to deliver, and act with urgency, definitely on the top priority projects. So on daily basis, we communicate with partners, we try to remove bottlenecks. We also on daily basis, besides to that part, we're thinking of improving our current workflows, looking at bottlenecks. Can we do this in a better way? Can we build a new workflow that we currently don't have? Can we look at getting things done in a more high throughput fashion? Because time is the only commodity we cannot buy. So to sum it up, on daily basis, we do science, we look at prioritization, there's a leadership role, making sure things are moving forward, and communication.
And I noted that you've co authored papers with both undergraduate and graduate students from schools like Gustavus Adolphus College. What advice would you give to those undergrads or graduate students who want to get involved with research that somewhat bridges that gap between academia and industry?
First of all, I'd like to mention that our experience with undergrad students and grad students, but specifically undergrad students, has been really great. We had interns coming to our labs for around 3 months. It's a 12-month internship at the different companies I worked for. And these students are very focused. They have a goal. They want to learn either a technique or learn more about industry or publish their first paper, and they do really well because they come with the focus, with the commitment to get things done. And for the many interns we had over the years, we did not have any intern leaving without publishing a paper. We had interns publishing 4 papers. We have multiple interns publishing their first author paper. So it was really great experience, and I always love these internships because it's a win-win for everyone, for the student, for the group. And I'm always interested in working with the students because it's really a fruitful collaboration between all parties. My advice for the students is to get involved early. Find a way to reach beyond your own institution. If you go to a conference, that's when you could communicate, could make connections with scientists and industry. And I have to highlight when you have a poster, that's really the natural opportunity to talk to scientists in industry. Make your intention known. If you have a scientist from industry stopping by asking you questions, ask them if there's a possibility for internships. Most of the big pharma companies have these internship programs. There's no magic bullet. You have to try applying to all of these positions. And also on the other side, we need to work more with professors, and when I say we, people in industry, science and industry, to also encourage them to help their students and encourage their students to apply for these internships, because it could be a life-changing experience. These students could go to doing their graduate studies with I would say maybe minimum understanding of what industry needs. A short internship in industry could really help them understand what the need is, and maybe select their PhD advisor or select the project they're working on, because the majority of the students in grad school are going to industry, not to academia. That's where the need is.
And that's certainly something that I would emphasize on my side in terms of industry, but the manufacturers of instrumentation is that I would certainly like to see more people like myself at those poster presentations, like at an event like ACS, one of the national
Mhm.
meetings. I know you've worked the expo booth during the day and you're tired, but there's a whole lot of value you can derive from going at night and talking with the people doing this research. And you might end up meeting your next great hire or someone who could really add to your organization.
Absolutely. Absolutely. 100%.
Now, one phrase I was totally unfamiliar with that I was hoping you could explain today is in silico modeling is a term that appears throughout your work. Could you explain that in plain terms and talk about why it's become so central to modern chromatography?
Of course. So when we're saying in silico modeling, pretty much we're making the computer do some of the experiments for us. So the way we do it is we start with a set of carefully designed experiments we run in the lab, usually around 9 experiments for chromatography modeling, and we feed it into the software. Now, the software will build retention models for chromatography, understanding how the retention time changes with composition, mobile phase composition, and how does it change with temperature. And we'll build a resolution map, we'll build a model where we could extract conditions under which we have the best separation. So it's a combination of computer power plus understanding the modeling part or how the retention models work, and few experiments from the lab which help us build these models. Now, there's if we want to do this in the lab ourselves, what I mean by that is testing all possible combinations. It could take forever. It's mission impossible. Now, with the power of the computer and the retention models, we could save all of that time and predict the conditions under which we have the optimum separation. Most of the time, we're looking at better resolution between critical pairs, which are co-eluting, or multiple critical pairs. So the computer will be able to do that job while we minimize the trial and error from our side. So it's more rational, driven, data-driven workflow.
You mentioned your use of resolution maps and how they can help you find the optimal separation. Are there any other details that might be helpful to someone who's only seen a resolution map by running a basic HPLC experiment in a teaching lab?
Sure. So in a teaching lab, we usually have a set of standards that are selected to have wide resolution among all the peaks. So you're really looking at one chromatogram. When we talk about these resolution maps, the best way to think about it is we're looking at every dot on that resolution map is a chromatographic condition. It's, let's say, gradient time and temperature. And for complex samples, we can't really run one condition and get all the peaks resolved. This is impossible in real life samples. So we have to run many experiments. If we're doing trial and error, it could be days, if not weeks, until we get all the peaks resolved. The resolution map, if we look at it, each dot is telling us a chromatographic condition. And the color indicates the resolution between the peaks. When we have blue color, means no resolution or minimum resolution. And the orange or red color indicates maximum resolution between the critical pair and the separation. So instead of randomly changing the conditions in the lab, we feed some data to the software which splits this resolution map. And our job is to go into these orange areas. Mainly, we focus on the largest orange area because that's the more robust area. And we extract condition within that area that fits our needs, most of the time, maximum resolution between the peaks of interest.
And I believe that your group demonstrated that only 9 runs can generate enough data to model an entire separation landscape. How does that compare to the traditional trial and error approaches people might be more familiar with? And how critical is that to you in a pharmaceutical setting where time is money?
Absolutely. Yeah. I have to be clear that trial and error is still used, especially when you have fit for purpose methods. There's no time to build these models, especially in the early stage where, again, speed is the key. We need a method. It's okay if the purity is plus minus 1%. So trial and error is still heavily used. Now, in later stages, it's really dangerous, let's say, to use that approach because, first of all, it could take long time to resolve all the peaks, number one. Number two, you don't really know if you're working at the local optimum because you may have multiple local minimum within that separation. When I say local minimum, meaning the combination of the conditions, let's say temperature and the gradient, where we have good resolution. So you may arrive to a good condition, but you don't really know if this is the best condition. And the only way to do it is with modeling, because modeling could explore all of that area simultaneously and point you out to the local minima. Now, the other thing I want to clarify is we always repeat that model helps you find the optimum conditions, but this is not the best way to think of the model. The model you build is a map. So you can always go to that map and make changes. I'll clarify two examples we used in the past. If a peak is no longer important, say you have an impurity you were able to purge during the process, you could go back to that map and delete that peak and now redevelop the method with no experiments needed. You just redevelop that method. Maybe you could create more resolution among the peaks, or maybe you could make the method shorter. There's another situations where now a new peak showed up. There's degradation happening, and this could happen when you put sample on stability. If you have that map, which acts like a database, you could go back and add a peak, and the software will help you spit out new conditions under which you have all the peaks resolved. So it's really the risk with the trial and error. You may get to good condition, but you don't know what you're missing. And if you need to make any changes, you're back to square zero. The other thing is any mistake in later stages with method failing during transfer or for any other reason, column robustness, is a very expensive mistake. You really do not want to push a method that you know will be used for the next 20, 30 years if it is not the best method. So it's a little bit of time investment ahead, but it saves a lot on the long term.
And I know that you've applied modeling tools in so many different diverse areas, whether it's small molecules, large biomolecules like proteins and vaccine, and tandem columns, and even 2-dimensional liquid chromatography. Is there a separation problem where in silico modeling doesn't work well yet, or where has it struggled the most?
First, I'd like to clarify when these models fail or the common causes when for these methods, for these models to fail. Then I'll mention a few challenging examples. So, in my opinion, we should use these models as a GPS. No, it's not magic. if you don't know where you're going, the GPS will not help you. Same with these models. You may want to make sure you have a robust method, robust model, and you know what the ultimate goal is. First of all, the model is only as good as the experimental input. You need to carefully design your experiments to cover as much space as possible without running into problems. I'll clarify a few technical things for colleagues who build these models. Any mistake in determining the dwell volume or determining the column depth volume, or the biggest one is not giving sufficient time for column equilibrium at the end of the run, could lead to a problem with the model. And by the way, when you feed the data to the model, when you compare the experimental to the predicted, it looks great. These errors in dwell volume, depth time, and equilibration time don't show up in the first experimental design where you have the 9 experiments loaded into the software. Usually they show up when you start predicting conditions outside of these 9 experiments, even within the design space. Because now if you look at the retention equations for chromatography, all of these factors play a role. So we need to be very careful determining the correct dwell volume, that time, and giving sufficient time for column re-equilibration. Now we have shown that 9 experiments are sufficient, and we've done it for different modes, and it was successful. I have to say, for example, for reverse phase small molecules, it's really easy. You don't even need the 9 experiments. 4 experiments would be sufficient, but the risk is you can always draw a line between 2 points. So you never know what's wrong. So that's why we suggest the 3x3. The challenges are more for large molecules, because large molecules are more sensitive to changes in %B and also temperature. And if you look at the linear solvent strength theory, we could see that the slope is really steep for large molecules when you look at log K versus %B. So any change in %B would give a lot of variability in retention time. So these are some of the challenges for large molecules. There's also other challenges. I would mention, for example, SFC. We tried multiple times. We got the help of one of the vendors also to try at one point, but we did not put enough time to build these models, but it's definitely very challenging. We have not been able to build a model for SFC successfully. The other challenge is having an autonomous method development, I would say, package or software, because you always need the analyst input. There's no software at this point which can really do the whole thing with no analyst input. So you could put your sample and optimize temperature and gradient simultaneously, and after some time you just get the method fully optimized. There's one software that can do gradient without temperature, but without temperature, we're missing a lot. I have to mention that I've seen some really impressive work at the HPLC conference, work by Bob Pirock, where they're looking at autonomous method development. Seems very promising. I have not used their software, but we have some really good steps in that direction. So to sum it up, I would say the large molecules present a challenge. Modes of chromatography other than reverse phase becomes more and more challenging, especially when you look, let's say, at hydrophobic interaction chromatography, with the complexity of the separation, the autonomous method development, and the SFC separations.
Imad, one thing that was very exciting to me about your resume was that we've spent a couple episodes devoted to some of the latest and greatest in GC, but we haven't done really anything in terms of two-dimensional liquid chromatography. It sounds intimidating. How would you pitch to an undergraduate who understands basic HPLC what 2D-LC really is and what problem two-dimensional liquid chromatography can solve that regular HPLC can't?
So I'll pitch it in the way I got interested in 2D-LC when I was doing polymer analysis. So in general, one-dimensional LC can answer one question, but if you have two questions in a sequence, you can't answer with one-dimensional liquid chromatography. You have two-dimensional liquid chromatography. So to give an example, if you have two copolymers, a copolymer is a polymer that has two different types of monomers. Let's call it A and B. You could have two copolymers of this similar molar mass, molecular weight, and when you run SEC, they would coat at the same time, approximately same size in solution. However, if you run the same sample with the two copolymers on an LC, driven by interaction now with the stationary phase, you could resolve these two peaks. So that additional dimension of separation gives you more visibility. And with let's say polymers, you have all types of different heterogeneities. You have molecular weight distribution. You have also the chemical composition distribution. You have other types of heterogeneity, chemical heterogeneity, sequence heterogeneity. So when you couple these two different dimensions, you have better understanding of the properties of the sample. So you're combining two complementary separations to answer questions that you cannot do with one dimension.
And you mentioned that the adoption of 2D-LC has been slow, partly because there is a very real steep learning curve. Do you foresee software developments or other tools potentially lowering those barriers to entry?
So I recall when I interviewed for the postdoc position with Peter Carr back in 2010, he told me we need 10 more years for industry to adapt using 2D-LC, and I believe he was correct. When you look at publications, conferences, you see a lot of 2D-LC being used, a lot of publications. I have to say what I've used to see in Pete's lab with the 2D-LC, three different computers, a helium tank, and three different software to control the system. Now it's a much more advanced system. The software is much easier to use, and it's definitely implemented more in different stages of drug development and it's in different industries for investigational purposes, for analysis purposes. Definitely, the threshold became much lower for using 2D-LC in industry, and the software is getting easier and better. In terms of modeling, we've published a couple of papers on using modeling for 2D-LC. The challenge with the models at this point, we don't have a software which can automatically extract conditions from two models simultaneously. So if I want to clarify that, if I'm running a 2D-LC experiment, which that was the scope of one of the publications, and I'm trying to find a condition for the second dimension where I could run it and I have separation of the peaks for every fraction coming from the first dimension. What we did is we built a model for every fraction in the second dimension. But the challenge is finding common conditions between all these different models. That's something we did by hand, going to the software, changing, making a change here, and going to the other model, make a change. So extracting information or common conditions from multiple models simultaneously is not something that's possible at this point. But once available, it will help a lot. We currently have a paper and review related to ability to extract conditions from different models and compare different models simultaneously and the importance of that. It will really advance that a lot. But overall, I see we're heading in the right direction, and we'll definitely lower that barrier a lot.
One thing I was especially excited to discuss today, because obviously green chemistry is a topic that we've covered on the podcast. You had a recent paper last year that mapped not just resolution, but also the analytical method greenness score across an entire separation space. And that's been described as the first. Why has separations field been slow to incorporate some of the sustainability thinking?
I think it's mainly for practical reason. In industry, we deal with very complicated samples. So we are trained to optimize resolution, sensitivity, robustness, turnaround time. This is the first thing we're looking for. Most of the time, sustainability is treated as secondary parameter we would look for. And once you have a good method, people are hesitant to touch and change because it could change selectivity. So that's one of the reasons. Now we're getting better in, I would say, changing this mindset by putting guidelines on what to use, what not to use, having different tiers for screening. First of all, start with this tier one. If we don't get a head start, go to tier two, and tier one would be greener than tier two. The other reason is there's also a mindset that if I'm doing analytical scale testing, it's not really very important to look at these parameters because it could be a few injections or so compared to purification, which is a lot of solvent usage, a lot of material analyzed. So the mindset is the sustainability is really important for prep, but not for analytical. But again, I think we will be better in taking that into consideration once we see more implementation of these tools into the software. So the vendor software, if we have these metrics embedded, that would really help, and that's when we need the vendors to help us with that. Maybe when you put the parameters, your gradient, your flow rate, etc., you get some metrics saying your greenness factor is this much, and the analyst could change few things and see how that factor could change, or simple changes, acetonitrile to methanol. How would that help? That will help build the mindset to start moving into building greener methods.
Yeah, that was the example that really stuck out to me, frankly, was when you replaced ACET with methanol in one of the examples and got a greener method that still worked. But you also found cases where the greener alternatives just didn't perform. How do you balance greenness against analytical performance in practice?
So I would say we cannot say green at any cost. We have to say we need to develop as green as possible methods while still fitting the purpose we're targeting. I believe this is possible using modeling. And as a follow up study for what you just mentioned, the paper we have in review right now, we actually used modeling to match retention times. So we have shown that using modeling, you could take two different methods, completely different selectivity. We've done it for formic acid TFA or acetonitrile methanol. And with modeling, we were able to match the retention time of different separations. And the difference between methanol, acetonitrile, or formic acid, or one column and another, even we've done perchlorate and TFA, which have really different selectivity. The difference was less than 5%. Most of the time was less than 2%. But a lot of the work was done manually, and we have a step-by-step process within that publication how we were able to do it with the model. Really nice work. I hope we'll be able to have that published soon. Again, the challenge here, as I mentioned in the previous question, is we don't have the ability to extract conditions from two models simultaneously. If we had that possibility, this would be relatively easy job where I could build the model with acetonitrile, build the model, and automatically find conditions in which we could match that retention time and get same selectivity. So I think it's possible. It's possible. We definitely have to push toward greener methods while still trying to meet the purpose of the method we're developing.
And one question I was very excited to ask you, coming back to something that the audience might be wrestling with, what's one piece of practical advice you would give an undergraduate who just ran their first HPLC experiment but really wants to get better at method development? What would you say to them?
Don't treat HPLC as a black box. That's my recommendation. And build a mental model of the separation. So when you're trying to change any condition, do it in a consistent way so you can understand how things change. If you're changing temperature, if you go up or down, try to understand how things move. So any change, try to understand how does that affect your separation. So don't collect random chromatograms. Also, I would highly advise, and I've done this myself when I first started working on method development, look at the LCGC articles, which we have a long series by John Dolan, I think from 1983 until 2000, maybe 16 or 17. I've read the whole series just by dedicating like 15 to 20 minutes every day, and these papers are really easy to read. There's also Dwight continued this series, really easy to read, and I know that's how John Dolan initially wanted these papers to be written, and they're great help. There's also a lot of resources. There's, which I recently known about, is a lot of presentations by someone at NIST, Sanders at NIST, Elaine Sanders, a series of presentations which really helps understanding the fundamentals and method development. So I would highly encourage people not to treat it as a black box. Don't worry, these systems are very robust. Don't have fear of changing things, trying things, and try to build that mental model where you could understand how things change when you change any parameter within your separation.
And finally, what's a problem in separation science in general that you think is still genuinely unsolved but is worth working on today?
I honestly think the main problem we have is we don't have enough students and we don't have enough professors. So we need more trained analytical chemists. That's the major problem. We have a huge shortage in the graduates who understand the fundamentals of chromatography. That's why I'm really happy when I see more professors in the field of separation science. We need those. We lost a lot of great professors, either retired or passed away in the last years or so. I think the other, aside from that, I mentioned about the challenge with the SFC separations. We need also autonomous method development with no analyst input. That would be really important because that will help people in industry a lot. The other thing which I think is important is the problem of predicting selectivity. I know it's easier said than done, but if there's a way, especially in high throughput environments such as Discovery, if there's a way we could look at the structure of a compound, let's say for chiral separation, and say on this column, using this condition, let's say on the IC column using methanol and DEA, I would expect the isomers to separate. That would be really the next huge breakthrough that could happen. I know some people have done some work on this regard, including Chris Welsh, and I think Bob Pirog is heading that direction. Dwight has done some work also in that direction. So that would be a huge breakthrough in the field.
No, I think that's a great answer. Imad, thank you so much for your time today. I think this has been a great discussion and so excited to share this with the audience and some really actionable items and topics for people to think about, especially the greener method development really stuck out. So thank you so much for your time, and I hope we can connect soon.
Thank you very much. Thanks for the invitation. It was a pleasure talking to you.